Let's talk of the MTHFR gene and does it affect egg quality and embryo development. So, in this study done in Korean women undergoing IVF and ICSI, the MTHFR gene was evaluated. It's an enzyme crucial for processing folate, a vitamin essential for DNA production and cell growth. Two common genetic variations, C677T and A1298C, can reduce enzyme activity, potentially impacting fertility. Researchers analyzed records of 173 women who underwent 301 IVF cycles who had undergone MTHFR genotyping and were taking folate supplements. Now, they compared outcomes such as number of mature eggs, quality of embryos across MTHFR genotypes. And what they found out that the results showed that women with this 677TT genotype, which had the lowest enzyme activity, has the lowest number of mature eggs, 71.4%, while those with 677CC genotype, normal activity, had the highest at 80%. Interestingly, the best rates of good quality embryos were seen in women with 677 CT genotypes, around 40%, higher than both 677C and 677TT groups. However, when it came to number of embryos suitable for transfer, there was no significant difference. What does it suggest? It suggests that the MTHFR genetic variations can affect early stages of egg and embryo development. However, with all participants taking folate supplements, the differences in embryos available for transfer were minimized. Now, this study highlights the importance of adequate folate intake for women undergoing fertility treatment, especially those with the MTHFR gene. Should we test it routinely? We don't know. This is one of the few studies which have just come up, but it may give us in the future some idea about how to improve egg and embryo quality. The study has limitations because one, it is retrospective, there's a lack of data on pregnancy outcomes, and it focuses on maternal rather than paternal genetics. All of those took the same dose of folate, so it's unclear whether higher doses would benefit those with low activity genotypes. But again, this gives us some idea that into looking at factors other than what we know regarding egg and embryo quality.